2020, a genetics lab in Warsaw. The latest results appear on the computer screen. 5,852 samples of maternal lines, 2,705 male Y chromosomes. All 16 voivodeships of Poland. The largest DNA study in Polish history. Scientists expected to see the usual European picture, a mixture of genes, the influence of neighbors, traces of migrations. But what appeared on the monitor made them reconsider everything. One mysterious haplogroup dominates with incredible force, 56.93%. No other concentration exists anywhere in Europe. This is not just a statistic. It is a boundary, a genetic line that divides the continent in two, East and West, Slavs and Germans. And this invisible boundary runs right through Poland, through every cell of the Polish body. Today, we will find out what lies behind these numbers and why they are rewriting the map of Europe. Today, we will discuss 4 key points that will turn your understanding of the origins of European peoples upside down. First, we will learn what the genetic boundary East-West is and why it runs precisely through Poland. R1A versus R1B, Slavs versus Germans, 2 Europes at a molecular level. Second, we will dive into the mystery of the origins of the Slavs. Did they arrive suddenly in the 6th century, as chronicles assert? Or had they been living here for millennia? Genetics provides a shocking answer. Third, we will examine the paradox of Polish DNA. How does a nation remain genetically homogeneous after 11 million displaced people, endless wars, and partitions? And finally, we will uncover the connection between the mysterious culture of corded ware pottery from 5,000 years ago and modern Poles, a thread stretched across centuries. And at the very end, I will tell you about one genetic marker that exists only in Slavs and appeared in the Bronze Age. This is archaeology written in DNA. Genetic analysis involves working with millions of data points. Every letter of the genetic code must be read without error. Every mutation, dated. Every haplogroup classified. This takes months. Computers work around the clock, processing terabytes of information, building phylogenetic trees, calculating statistical probabilities, comparing sequences with thousands of samples from European databases. The team of scientists awaited the results. Poland is the center of Europe, a territory where historical paths converge. Trade routes passed here, cultures mixed here. Different ethnicities coexisted here for centuries. And now the computer completed the final calculations. Numbers appeared on the screen. Percentages. The distribution of haplogroups. Phylogenetic connections. Everything that scientists had sought for months. A picture of the Polish gene pool. Complete. Detailed. Ready to answer questions about the origins of the people. Did you know that Poland is home to a population with the highest frequency of one Y chromosomal haplogroup in all of Europe? This is Haplogroup R1A. 56.93% of all Polish men carry this genetic marker in their cells. Here's an astonishing fact. When geneticists analyzed samples from all voivodeships in the country, they found something incredible. This figure exceeds that of any other European nation. Just imagine. In neighboring Germany, R1A makes up only 17.9%. In the Czech Republic and Slovakia, it's 36 to 38%. Even in Ukraine and Russia, the figures are lower, from 37 to 55%. But in Poland, this haplogroup reaches an absolute maximum. It is a genetic fingerprint that distinguishes Poles among all the peoples of the continent. But where did this mysterious R1A come from? Professor Łukasz Grochowalski from the Center for Forensic Genetics at the Pomeranian Medical University, who led the large-scale study, asserts, Haplogroup R1A is a defining marker of Slavic Y chromosomal heritage. Its origin is related to the Indo-European expansion from the Eurasian steppe about 5 to 10,000 years ago. The mechanism is simple yet astonishing at the same time. The Y chromosome is passed exclusively from father to son, practically unchanged over hundreds of generations. It is the perfect tool for tracking paternal lines through the centuries. The research identified the highest frequencies of R1A in central regions of Poland. In Łódź Voivodeship, 68.14%. In Kujawia, Pomerania, 63.38%. Absolute records for Europe. The calculations show that the subclades of R1A date back to 1,600 to 2,900 years ago. But there's a nuance. Ancient samples from the Volga-Oka region indicate an even earlier origin. Traditional history states that the Slavs appeared in Central Europe in the 6th century AD. The Great Migration of Peoples. Byzantine chronicles first mention the Slavs during this period. Procopius of Caesarea writes about them as a new people who came from the east. But genetic data tell an entirely different story. The subclades of R1A found in modern Poles date much earlier, long before the 6th century, centuries earlier. And the roots of these genetic lines stretch even further back in time, to an era when Rome was still an empire. This creates a paradox. The Slavic genetic line was present in Central Europe long before Byzantine historians documented the arrival of the Slavs. Who were these people? Where did they come from? And why did their descendants end up here in Poland? The answer lies in archaeology. By the way, a fun historical fact. In 2010, geneticists published a study that brought together two sciences. They compared the geographical distribution of the R1A1A7 M458 subclade with archaeological maps of Europe, and made a shocking discovery. The range of this subclade nearly perfectly overlapped with the territory of the ancient Corded Ware culture, an archaeological culture from the Bronze Age, existing 5,500 to 4,500 years ago. In the paper, this was described as a remarkable geographical correspondence. It turned out that the modern distribution of Slavic genes replicates the map drawn by archaeologists from pottery shards that are 5,000 years old. Genetics and archaeology were telling the same story, a story that the bearers of the Corded Ware culture were direct ancestors of modern Slavs, and the highest concentration of their descendants turned out to be right in Poland. This brings us to the next question. Now let's explore what was present in the territory of Poland before the arrival of the R1A bearers, before the Bronze Age, before the Corded Ware culture, before the Indo-European expansion. Are you ready for a revelation that takes us back 10,000 years? In second place by frequency in the Polish gene pool is Haplogroup I, 15.71%. Almost 16% of modern Polish men carry this ancient European line in their cells. Haplogroup Y is not Slavic heritage. It is something much older, Paleolithic, a trace of hunter-gatherers from the last ice age. People who lived in Europe over 10,000 years ago, when glaciers were just beginning to retreat, when mammoths still roamed the tundra, when there was neither agriculture nor livestock, only hunting, gathering, and survival. And these ancient Europeans did not vanish. They were not wiped out by incoming migrants from the east. They became part of the people we call Poles today. Here are some concrete figures that create an astonishing picture. The study of 2,705 Polish men showed that Haplogroup Y is distributed unevenly across the country. And this distribution tells its own story. Western regions of Poland. Lubusz Voivodeship, 25.47%. Lower Silesia, 23.38%. These are the highest figures. Almost a quarter of the population carries an ancient Paleolithic line. But now look at the eastern regions. Podlaski Voivodeship, 19.9%. Also high figures. Now let's compare with neighbors. Slovakia, 27%. The Czech Republic, 28%. Germany, 23.6%. In all of Poland's western and southern neighbors, Haplogroup Y appears more frequently than the average Polish figure of 15.71%. But Lithuania has only 10.4%. 1.5 times lower than Poland. What does this mean? Poles are at the border of two ancient genetic zones: Western European, where the Paleolithic population has remained more intact, and Eastern European, where it is less so. Imagine the territory of modern Poland in the period after the glaciers retreated. People already lived here, bearers of Haplogroup I Paleolithic Europeans. Their descendants constitute today 1/5 of the Polish gene pool. Then came the Neolithic era. The greatest revolution in human history began, the transition from hunting to agriculture. And this revolution did not come by itself. It came with people. Agricultural tribes moved from the south through the Balkans. They brought with them a completely different way of life: agriculture, animal husbandry, permanent settlements, and different genes. In the modern Polish gene pool, haplogroups E, J, and G of Near Eastern origin are present, markers of Neolithic farmers. E, 3.84%, J, 3.22%, G, 1.22%. In total, 8.28%. Two populations met in Central Europe: Paleolithic hunters and Neolithic farmers. Instead of war, there was a merging, a mixing of genes, the creation of a new gene pool uniting the ancient Europeans and the newcomers from the south. But here's what truly blows the mind. Scientists applied the AMOVA method, analysis of molecular variants. This statistical tool shows how genetically different various regions of a country are. The results were staggering. For the Y chromosome, 99.25% of variation occurs within the population, only 0.75% between voivodeships. For maternal lines, it is even more striking: 99.78% within the population, just 0.21% between regions. What does this mean? A Pole from Warsaw is genetically almost identical to a Pole from Krakow or Gdansk. Regional differences are negligible. It is a single genetic mass from the Baltic to the Carpathians, from the odor to the bug. Poland is one of the most genetically homogeneous nations in Europe, despite its complex history. Let's start with the revelation that shows Poland as a contact zone of two worlds: Haplogroup R1B, 14.9% of the Polish gene pool. This is a Western European line, dominant in Germany, France, Spain, and Britain. In Germany, R1B reaches 38.9%. In the Czech Republic and Slovakia, 20 to 25%. In Poland, just over 14%. But it is still a significant share. Every seventh Polish man carries a Western European genetic marker. Interestingly, R1B is distributed evenly across Poland. There is no Western gradient, no concentration near the German border. This indicates one thing: Western European genes entered the Polish gene pool a long time ago, possibly in the Neolithic or Bronze Age, and have spread evenly since then. Poland is the border between two genetic worlds, the meeting point of R1A and R1B, Eastern and Western Europe at a molecular level. A fun modern fact: after World War II, Poland experienced unprecedented population displacement. More than 11 million people were relocated or displaced. The country's borders shifted westward. Entire cities changed their populations. Germans moved west. Poles came from the east, from Crécy, eastern territories that were ceded to the USSR. From the Lithuanian SSR, about 200,000 Poles were relocated to Warmia-Masuria voivodeship. Western Pomerania and other regions received people from different parts of pre-war Poland. Such large-scale mixing might seem to have destroyed the genetic structure of the nation, but genetic analysis shows the opposite. Homogeneity has been preserved. Why? Because genetically similar people were resettled. Eastern Poles are indistinguishable from Western ones at a genetic level. Poles from Vilnius had the same genetic profile as Poles from Poznan. History redrew the borders, but genes remained and told the truth about the unity of the people. Now let's look at the fourth largest haplogroup in the Polish gene pool. N, 4.29%. A small fraction, but extremely illustrative. This is a Baltic-Finnic Uralic line, a marker of the peoples of northeastern Europe. For comparison, in Lithuania, haplogroup N reaches 44.9%, almost half of the Lithuanian population. In Poland, just over 4%, a difference of more than 10 times. But even this small percentage is enough to see traces of historical connections. The Polish-Lithuanian Union existed from the 14th to the 18th century. 500 years of shared statehood, dynastic marriages, migrations, trade between regions, military campaigns where Poles and Lithuanians fought side by side. All this left a genetic mark. 4.29% is a memory of the time when the Commonwealth unified the Slavic and Baltic worlds under one crown. A story that gives you goosebumps. In 2019, an international group of scientists completed an analysis of maternal lines in the Polish population. Mitochondrial DNA passed exclusively from mother to children. 4 main haplogroups made up 82.38% of all maternal lines. H, 43.42%. U, 20.6%. J, 9.77%. T, 9.13%. But the main discovery lay in the details. Scientists found subclades specific to Slavs. H5A2, an almost exclusively Slavic haplogroup, dated to 2.5 to 5,000 years ago. U4A2, U5A2A, U5A2B, 6 to 7,000 years ago. And here's what is astonishing. The distribution of these maternal subclades coincides with the distribution of the paternal subclade R1A1A1M458. Maternal and paternal lines expanded together. This was not a conquering expansion of men. It was the migration of entire families. So what have we learned about Polish DNA? Poles are the result of 3 ancient waves of migration. The first, Paleolithic hunters with haplogroup I, 15% of the modern gene pool, the earliest Europeans. The second, Neolithic farmers from the Near East, haplogroups E, J, G, 8%. The third, Indo-European pastoralists of the Bronze Age, haplogroup R1A, 57%. 3 waves created a unique genetic profile. Poland turns out to be on the border of 2 worlds. Here, R1A reaches its peak for all of Europe. Here, the Slavic East meets the Germanic West, at a molecular level. But what is most striking is genetic homogeneity. More than 99% of variability is within the people. 11 million displaced after the war did not destroy the unity. Poles remain a single genetic population from the Baltic to the Carpathians. Poland is not just a country on the map. It is the genetic maximum of the Slavic world, the point of the highest concentration of ancient Indo-European lineage. Now think about this. If the genes of the bearers of the Corded Ware culture, who lived 5,000 years ago, still dominate the Polish gene pool, what does that say about the nature of national identity? We tend to think of peoples as constantly changing communities, conquests, migrations, cultural mixing. But genetics tells a different story, a story of remarkable stability amid historical chaos. A modern Pole from Warsaw is genetically closer to a person living in the Bronze Age in the same territory than to their contemporary from Berlin or Paris. 5,000 years of history, wars, empires, religions, have not erased this genetic imprint. Borders on maps have changed hundreds of times. States have risen and fallen. But genetic boundaries remain stable for millennia. Poland stands at the confluence of 2 Europes not by the will of politicians, but by the will of ancient migrations, recorded in every cell. Perhaps what we call national identity is not just culture. It is also an echo of the past and the present. And now about what I promised at the beginning, about the genetic discovery that overturns conventional understanding of the origins of the Slavs. In 2013, an international group of geneticists completed a large-scale study. They analyzed complete sequences of mitochondrial genomes of Slavic peoples. seeking an answer to the main question: where did the Slavs come from? The results refuted the traditional theory. The scientists found Slavic genetic markers dating back long before the early Middle Ages, thousands of years earlier. The authors of the study formulated a revolutionary conclusion. The data indicate that the ancestors of the Slavs were autochthonous peoples of Central and Eastern Europe, rather than early medieval invaders. The autochthonous hypothesis. The Slavs did not come from somewhere external. They formed here in Central Europe over thousands of years, from a mixture of Paleolithic hunters, Neolithic farmers, and Indo-European pastoralists of the Bronze Age. By the 6th century, the process was complete, and the Byzantines merely recorded what had been shaped over 3,000 years. What do you think? Do these discoveries change your understanding of where European peoples came from? If the autochthonous hypothesis is correct, then the traditional version of the Great Migration of Slavs in the 6th century turns out to be a myth. Should we rewrite school textbooks, adjust academic theories? What is a more reliable source of information about the past? Byzantine chronicles written by people with their own agendas and biases? Or a molecule of DNA that objectively records events over millennia? And here's a practical question. Perhaps some of you have taken a commercial genetic test? Have you discovered Slavic markers? A high percentage of R1A? Or maybe unexpected admixtures that family legends did not mention? Write in the comments. This is not just an academic discussion. It is a question of how we understand our own history and identity. And finally, the 2020 study is the largest in the history of Polish DNA research. But this is not the end. It is only the beginning. Sequencing technologies improve every year. What cost millions and took months 10 years ago now can be done in days. And in 20 years, it will be even faster and more accurate. Future geneticists will analyze ancient DNA samples from Poland's archaeological sites, from the graves of the Corded Ware culture, from Neolithic settlements, from Paleolithic campsites. They will read the genomes of people who lived 2, 3, 5,000 years ago, and compare them to modern Poles. They may discover subclades that we cannot see today, details of migrations that remain a mystery, connections between peoples that we do not suspect. DNA preserves more information than we are currently able to read. Each cell is a library of history, and we have only just begun to explore it. The history of the Polish people has not yet been fully told.